Hello, everybody. Thank you for attending our VALPRO Path webinar on creating plant protein value chains for faba beans. Today we have three of our partners from the VALPRO Path project who will be presenting the findings from the project: Dr. Sheila Alves, Professor Mark Fenelon, and Dr. Eimear Gallagher, with Dr. Richard Lynch, presenting just a short, brief explanation in the beginning about the VALPRO Path project, and myself, Dushan Chines, who is one of the communications managers for the VALPRO Path project, will be moderating and so forth. I will give now the word to Dr. Richard to give us a few words about the VALPRO Path project, and uh, we will go from there. Thank you very much, Dr. Richard. You have the spotlight.
Thank you very much, Jason. This webinar is brought to you by a European-funded project known as VALPRO Path. It's funded under Horizon Europe, received 10 million. It began in September of 2022 and will finish at the end of 2026. There are a number of partners involved, about 22 to 23 different partners, with a broad mix across industry and research and so on. But the overall objective of VALPRO Path was really to look at protein crops that were being grown currently in Europe, and to see if we could generate high value business models for those crops so that they would be maintained in terms of production rates and so on across Europe. So the aim was to look at the main principle, I suppose, of the project was to look at the right crop. So in terms of the right variety of the legume that's been grown, that it's grown in the right place, right location, and then it's generated into a product that's delivered to the right marketplace. The five main crops we had a look at in the project were lupin, chickpea, faba bean, field pea, and peanut. And we also looked to a small extent on soybean. And I suppose the main questions that we tried to address in the project were ones that crossed the value chain the whole way through, from grower, processor, the whole way through to the end product or end application of that crop. So the grower would have specific questions around the return they would generate from their crop, and what is the best crop or variety to grow in that region to maximize yield and quality. Then the processor needed to know whether or not it was worth investing in a processing facility to actually take in this raw crop and generate value from that. So they wanted to know, was there raw material available, and was there sufficient technologies available to process that crop? And then for the end food manufacturer, was there a sufficient amount of crop processed ingredient available to generate their end product? And then a consistent supply of this raw material is always important to maintain their business model. So today's webinar is specifically focused towards Faba Bean, and we're running a series of webinars in the project. Today is Faba Bean. In 2 weeks' time, we'll cover chickpea, and then we'll cover lupin and peanut and so on. But specifically, we wanted to focus in on the research that's been conducted on Faba Bean. Why? Well, Faba Bean, as we know, is one of the oldest cultivated crops globally, and it thrives very well across different diverse climates. So it's a highly versatile crop and can achieve very good yields, specifically in northern climates such as Ireland and Denmark, two of the countries involved in the project. Its primary uses globally would be across Asia and Africa for human consumption, so as raw or green bean. But in Europe, it's been used to substitute, I suppose, or to improve the level of protein that's been given as a high value protein feedstock for livestock in Europe. Its benefits beyond that are that it's high in protein, 32, I suppose, to 38% protein, depending on variety. And it's very strong in terms of its carbohydrate and fiber profile. Contains many beneficial vitamins and minerals as well. It's very beneficial in terms of its place in a crop rotation. So it's a legume crop in terms of it fixes nitrogen freely available from the atmosphere, so reduces the need for synthetic nitrogen fertilizers. It also reduces the level of plant protection products that are needed in a crop rotation by breaking disease and weed cycles, and it improves soil structure and biodiversity of the crop. So overall, it's a very beneficial crop to both human, to the actual cropping system, and to animals itself. In terms of the crop, there's lots of opportunity around it. In the EU, the cropping area for legumes has grown steadily over the last decade, And this is particularly due to the fact that there's a deficit in terms of high protein feedstock available for our livestock sector. So there's been some coupled policy supports that have been put in place by the European Commission, such as the protein aid scheme that's seen in Ireland and so on. And that supports the production of these legume crops. So there is a large growth in this market based on that and also the demand from consumers for more plant-based products. However, they're still very underutilized in terms of their place in a crop rotation. And the reasons for that are around the variability that's perceived in yield that can be obtained from these crops. There's been a lot of research done on this, and a lot of the yield variability that's seen has been related directly through the knowledge of the grower and the producer of that crop. So because it's such a novel crop in some aspects, it needs a lot more research in terms of the advice and knowledge transfer of how to actually grow and manage that crop to produce high enough yield. It's also considered to be a not so profitable crop in comparison to your mainstream cereals. So there is a potential to improve the profitability of this by bringing it beyond the feed sector to the food sector, and generating some of that added value. So today's webinar is really based on addressing those core issues around Faba Bean. It's finding the right variety that grows in the right location, then trying to bring that crop then further down the value chain to add more value to this through processing, and then further on finding a food or a good end application for this crop for consumers to take up. So without further ado, I'm going to pass over to our first speaker, which, despite the name being Eimear Gallagher at the bottom of our screen, is actually Dr. Sheila Alves from Teagasc Park. She's a crops research scientist and has many years' experience in growing and cultivating and trialing these crops. So, Sheila, the floor is yours.
Thank you, Richard. Yes, apparently there's a duplication here of Eimear Gallagher. So Eimear, don't know why it's happening. I'm just going to share my screen. Oops. Share and share. And are you seeing my full screen now? Perfect. Okay, so my name is Sheila Alves. I work in Teagasc with Richard and Eimear and Mark, and I work in the crops department. So I focus on agronomy of protein crops. In terms of this project and of Faba Bean, the main focus, as Richard said, is looking at the right location for the right variety for the location. So, but also looking besides yield to the application. So when we talk about agronomic practices to optimize or stabilize yield, we are talking about variety here in blue. But we are also talking about all these parameters in these brown boxes: seed rate, soil fertility, weed, sowing depth, time of sowing, establishment system, that will actually determine the plant population and the plant development. There are variables that we cannot control, like temperature, soil radiation, water deficit, etc. So this is the location part. So we optimize the brown boxes according with the location, according with the non-controllable variables, sorry, and those will impact the yield. Variety by itself also has a huge impact in yield, but also in application. So for food, for feed, and other applications. So it's not only selecting a variety that yield well in Ireland, but also yield well for what market. So what characteristics are we looking in these varieties to produce the best bread, produce the best meat alternative? So we basically do two different investigations. One, we study different varieties in different climates, and the other was we study a selection of more than 100 breeding lines to see that were grown in the same place, to see how they vary in terms of characteristics for potential food applications. So in terms of the trials done in different locations, we did a trial in 2024. So we saw it in spring, 2 locations, Ireland and Denmark, and 2 varieties, Lynx and Louis. The difference between Lynx and Louis is that Lynx is a standard variety with a standard grow habit, while Louis is a fast-growing variety that is very determined. So it doesn't tend to grow shoots besides the main stem. And now some of the results in terms of yield. In general, in 2024, the yields in Denmark were lower than Ireland. Interestingly, in both countries, Louis performed better than Lynx, but this trial was actually sown late, and that favored the fast-growing variety. In terms of composition, it's interesting to see that Ireland had a bit higher protein content, and the lower protein content in Denmark was due to an increase in the starch content. Also, Louis was a bit higher than Lynx in Ireland, but the opposite happened in Denmark. So there's a trade-off that is environmental related too, not only varietal, but environment. The second study, when we look at 1,000 grain weight protein, globulins, and albumins in a panel of 119 breeding lines and 4 varieties, that was given by a breeder. So all of them were grown in the same season, the same field. So the only variation potentially we have here is genetic variation. And why are we looking for these particular characteristics? According with the food scientists, high globulin improve emulsification for plant-based meats, gelation properties for meat analogs, and foam for bakery and desserts. High albumin, on the other side, is associated with better digestibility, an increase in sulfur amino acid content, and improved amino acid balance. So, according with what we intend to use the protein from the crop, we can select the variety too. So it's beyond we yield here. Okay, so this is the results for the 1,000 grain weight. You have some pictures here, potentially not so good quality, but I just wanted to show you that there was a difference in shape, in color, in size in the collection. In terms of 1,000 grain weight, we went from a value below 400 grams per 1,000 seeds up to 800 and something grams per 1,000 seeds. So we actually have in this collection minor, equina, and major, which were the classical division of the Faba bean in terms of 1,000 grain weight. In terms of protein content, we also saw a big variation. It varied between 22 to 32, and this is using conversion factor from nitrogen from Dumas method to protein content of 5.4. So it's even a bit below compared to the standard of 6.25 usually used. So we have variation in this panel in 1,000 grain weight in protein content. So what about the globulins? Just presenting here some varieties of the collection, just to show the variation we found to date. And you can see that globulins vary from 75% of the protein content up to above 90% of the total protein content. This means that indeed the globulin content is as a genetic component, and it's correlated with the total protein content, as you can see in these graphs. So the lower protein content varieties are also the lower varieties in terms of globulins, and the higher protein content correspond to the higher globulin content. Albumin was not correlated with total protein, but it was correlated, not significant, but it was correlated with, inversely correlated to globulins. Another aspect that faba beans are becoming of interest is to support other crops in intercropping, in an intercropping scenario. And we did in VALPRO Path a lot of intercropping with legumes, so faba beans and peas. And the role of the faba bean here was not to increase the productivity, was not to increase the protein content in the intercrop mixed harvested. It was to support the peas, to decrease the lodging. Ireland has a high, and Denmark too, have high yield potential for both crops, which means peas become very heavy at harvest, and rain and wind can make them lodging suddenly. And there's a number of reports of crop loss. With the intercropping, you can see here the sole crop. This is just an example. It's an old trial we did. The sole crop of peas in the 16th of July, it was completely lodged. In August, the pigeons have attacked. We lost all the crop. The intercropped plots in the 16th of July were still standing, and in the 8th of August, they were still continue standing. But what's important here to see, to think is, okay, we have now increased the potential to produce peas in Ireland. Now we have, but we also have a mixed crop now. So what should we do with that? And now I leave that to Eimear, because she will continue to talk later about this. That finishes my part.
Well, thank you, Sheila, for a wonderful presentation. Now we go to Dr. Mark Fenelon to continue. Dr. Mark, are you ready?
Yes, thanks, Dusan.
Yes.
Okay, can you see that okay? Dusan, is that okay?
Yes, we can see
Yes.
the presentation, yes.
Very good. Okay, so you're all very welcome. And I'm going to follow on from Sheila now and pick up on what Richard has just explained earlier about how to create value from faba bean protein. And what we're doing here really in this part of the project was to create ingredients. So what we're talking about is creating a protein isolate or concentrate from the faba bean. And from the work that was done at the production level, then we monitored the different composition of faba beans, and then we looked at how much protein ingredients, starch and fiber ingredients can we get from them. So our focus here was on concentrating on innovative technologies then for extraction of protein from the fava bean. So that's what I'm covering here now. So maybe just to set the scene. So I suppose we looked at lupin, faba, pea, and chickpea, and obviously the focus here today is on faba bean. The ingredients then we looked at were protein concentrates, isolates, starch, fiber, and other bioactives, but I'm going to just cover the concentrates and isolates here today. And some of the processing technologies then I'll cover today is the dry and wet fractionation, hydrodynamic cavitation, and also the wet milling and a novel drying technology that we use as well. So we did establish pilot processes, and the reason for this then was obviously for our partners and for others to use and to develop the processes and to make them as energy efficient as possible. We looked at LCA analysis with University of Athens, and also a nutrient tracking model then from farm to the finished product we also looked at. So I'll talk a little bit about that as well. So that's just to show exactly how we can monitor the yield from the farm and how much ingredients and so on we can get from them. And of course, the carbon footprint here then is one of the key metrics as well. So I think if you look here at the picture in the middle with the fava bean protein isolate, this is ultimately what we're trying to get. So back to Richard's point again about adding value. So how do we do that? Well, we do it by producing high value protein ingredients. So this is above 85% protein we're talking about here. And you can see the fava bean on the left of your screen as you look at it, and the hulls and the dehulled fava beans down on the bottom. But it's really about how do we get this protein? And why would we want to do that? Well, it gets you into these value add areas then. And these are examples of protein-based formulations or lifestyle beverages. And they're in the areas of sports, nutritional, medical health benefits, infant growing up and adult health food formulations, also energy and diet formulations. So the key to this then in any protein, whether it's dairy, plant, or even marine, and while we're focusing on fava bean today, it's the same functional characteristics that are required, which is a good amino acid profile, a high protein quality, and then it fits the regulations, and obviously that it's cost effective to actually use these ingredients as nutritional bases to create other foods and formulations. And we do take into consideration here then, of course, the functional and the processing stability of these ingredients that we produce. So this is very important because when you make a finished powdered protein ingredient, you want it to be soluble and stable in the new formulation that you've created from it. Okay, so two examples of processes that we developed within VALPRO and have studied is the wet fractionation and the dry fractionation. So just to explain the difference between the two. So with the wet fractionation, we're talking about milling fava bean into a powder. We then use an alkaline solution. We spin out the insoluble fibers. We use acid precipitation then to take out the protein. And that's a bit like if you put lemon juice or enough of it into milk at home, you know, you can precipitate out your proteins or make a yogurt or so on. So basically we're doing the same thing with the protein in the fava bean, but we're using a mechanical separator then to spin it out and to produce an isolate. So the fibers and the starch don't spin out, they don't aggregate in the same way as the protein. So this is a well-proven technique for taking out the protein then off your fava bean and producing a value-add ingredient. And this differs from the dry fractionation on the other side of the screen, where we produce a powder, a fine powder, and then we use a cyclone to separate these based on density. So the protein-rich fractions and the carbohydrate-rich fractions will have different densities, and hence they'll separate differently. The big difference, though, is that the wet fractionation process gives you a protein content of greater than 85%. Where the dry fractionation is 60%. There's no water added. It's a simpler process, but you don't get the same concentration. And you also get a coarse fraction then of starch and fiber as well. And maybe just to set the context then, just to re-emphasize the point, if you look at this graph here, you can see then that the hulls, the flour, the fiber extract, and the protein isolate, you can see there we have in the protein isolate, which is the powder, we have now 85% protein in it. So that's a very valuable ingredient. So this is how we add value then. Now, obviously, I should mention as well, maybe not as much covered in this presentation, but we're also very interested in valorizing the starch and fiber fractions as well, which is very important to make it the economics of the business model work. So that's something we're also studying, but I'm concentrating on the protein here today. And these are just 3 examples of isolates that we made. And I should point that chickpea maybe not characterized as an isolate because it's 72% protein. If you look in the table here in red, The reason being is the fat content is higher, so we have to add another step to remove the fat in the chickpea. But from the Faba, you can see it's a 90% protein from the process we used, and this is a very good functional and nutritional ingredient. And obviously lupin then as well is 82, so again, close to an isolate. So some technologies then that we use. So I suppose one of the challenges with plant-based is that you have to, first of all, get them into solution. So the beans are obviously not in solution. So we do this by using a wet milling approach. And this technology then, we produced a coarse milling effect and then a fine milling. And then we could use our alkaline solution, our acid solution, and separate our protein. So one of the technologies then we use was this pumping system you can see in the top, and it produces this fine milled solution then, which we can then apply and separate the protein out of. So this is an important step. So as opposed to something like in a dairy where it's already liquid, you have to do this with the plants up front. So we studied this extensively through VALPRO. And you can create a powdered end straight from the milled material, or you can go on to produce the isolate that we spoke of. And I'll just play a video here now, and you can see. So you can see then where the fava beans are added into the milling system at the bottom, recirculated, and this gives us the coarse material that we've seen on the previous slide. Excuse me. Another technology that we looked at to improve the solubilization of our flour was hydrodynamic cavitation. So again, it creates cavitation, this particular pump does, at the center of the pump, which creates localized high shear phenomena, which causes our powders then to go into solution. So again, we did this for two reasons. One, to make sure that our fava flour will get into solution, but also then to obviously produce, to increase the protein yield that we got. I just showed you two small videos there, and the difference between the two, you can see one is liquid and one is the gel. The reason is because we changed the speed of the pump. So we introduced more cavitation, which introduced gelation, and then we get a different functionality within our final powder. So again, the reason for doing this was so that we produce a liquid which would not separate while used in downstream processing, and also then we also improved our protein yield that we could get from this particular material when we used it for producing the protein isolate. So again, an interesting technology which on two fronts helped us up front while producing our isolate. Okay, so we concentrated on wet fractionation. This is dry fractionation now. So I put up the slide we shown earlier, but really we're focusing here on two dry fractionated fractions. And maybe draw your attention here to the protein content of the fava flour, the protein concentrate, and the fava starch. So the coarse fraction, which comes out the bottom of our cyclone then, which is different density from the protein concentrate, which comes out the top. You can see very clearly here we have concentrated the protein in the fava concentrate. So it's 58% protein, as opposed to 27%, which was the original flour, and then the fava starch, which is 15%. So we've created a concentrate by not adding any water, any acids or alkaline solutions, but it's a much lower protein content, but it does have very interesting uses as well. So it's a different ingredient. So this is something we studied in VALPRO, and we have looked at the functionality of this in detail. And this graph here just shows you now how you can use the different functionalities of these ingredients to produce different products. For instance, the orange line here, which shows that while you increase the temperature, you end up with a much more gel-like structure, which can be used for solid-like foods, baked foods, etc. Whereas as the fava protein concentrate, which is the green line on the bottom, can be used for making things like nutritional beverages, which I'll show in the following slides. And then the middle there, we have the flour itself. So by dry fractionation and separating and making a coarse fraction, which is enriched in starch, or a protein fraction, we can produce different ingredients, different protein contents, but also different functionalities for different uses in different foods. So this was something, again, and there's a whole range of products that we have looked at within the VALPRO project. So then the next technology we looked at was an innovative drying technology. So it's a high shear turbulent drying system. And you could use this for drying powders, but you can also use it for drying the fiber and starch component. So I spoke about separating the protein, but also the residual biomass, which is the starch and fiber, can be dried on this system. So it gives you an opportunity then to produce powders and to increase the value chain, obviously, substantially, because you're using the entire Faba Bean. So this is something, again, which is core to VALPRO. And the reason you would do this, obviously, for microbial safety, also to produce a powder and obviously to increase the shelf life of your powders and to enhance the product texture and appearance as well. So again, this technology worked very well, and only in the last couple of months have we produced some nice starch fiber ingredients as well, which are complementary to our protein ingredient that I showed earlier on from the wet and dry fractionation. This is an example of some of the powders then, so you can roast the powders and get nice textures and flavors. You also substantially reduce the microbial load. And by doing this then, you can use these ingredients in some very high-end nutritional beverages as well, and further increases the shelf life. And it's very important when you're using these flours, this Faba flour, as a nutritional base then for other foods. So it was an interesting technology and gave us an option to produce a number of ingredients from the Faba flour or indeed from the protein ingredients or starch fiber ingredients. Finally then, the nutritional beverages. So we're back now to the protein ice that I spoke about. And you can produce very novel powders then, or ingredients, liquid concentrates, which can be turned into a ready-to-drink beverage, which you can see here. So we did this in VALPRO as well, and we built a number of formulations, mostly in the growing up adult type formulations, by using the protein isolate, the Faba protein isolate then, with some good fats and carbohydrates to produce some interesting novel beverages. So again, this was work that was carried out during VALPRO. Finally then, just to show some of the ingredients or some of the foods that could be produced with the non-protein side, which is the starch side of the starch coarse side of the dry fractionated Faba bean. And these are examples of a bread-like structure, very successfully produced with the low protein, high starch Faba fraction, again to show that you can use these the byproduct stream from the protein isolate production or the dry fractionated production for other applications. So again, this is something we've worked on. And then very finally then, these are meat crisps. Hopefully these are working okay and you can see them.
Yes, they're working, Mark.
Okay, so with the meat crisps then, what we did was these are very popular in Asia. So we used the dry fractionated starch enriched fraction, mix them with meat, and then you can see then when you put them to fry them, they turn into these crisps, which are very popular in Asia. And there's a whole range of different products then that we have been evaluating in this space. But again, it comes back to the functionality of the starch in the starch enriched fraction, and it's a nice ingredient which we're evaluating then in a range of applications through VALPRO. So it shows how you can add value from ingredients. So again, I think this is my final slide, but just to finish up then, back to the original comments made by Richard. How did we add value? We did it by producing ingredients, protein ingredients, starch ingredients, etc. And then we looked at obviously their carbon footprint, their functionality, nutritional value, and also their applications. Okay, so thank you for your time.
No, thank you, Professor Mark. Again, we have learned again so much, and we continue to work through the VALPRO Path project. Now we go to our final presenter, Dr. Eimear Gallagher. You may start when you're ready.
That's great. Thank you very much. Can you see my screen okay?
Yes, we can see it.
Great. So hello, everyone. My name is Eimear Gallagher. I'm a researcher in Teagasc Food Research Centre in Ashtown. So I am concentrating on food applications of intercrop fava beans and fava beans on their own, and in particular bakery applications. So as Sheila and Richard mentioned intercropping. I'm not going to spend too long on the slide. Intercropping, as we now know, is when we grow two or more different crops together in the same field over one growing season. In this instance of our intercropping trial, and where I am interested in, Faba Bean has been used to intercrop with pea. As we know, Faba Bean have strong stems, and they're able to reduce pea lodging, making harvesting more easy, and decreasing the damage to grain once harvesting. It also can help with giving a higher yield of crops. As been mentioned already, both faba beans and peas are legumes, and they fix atmospheric nitrogen, so there is a reduced need for artificial fertilizer, and all this increases the yield of productivity of the intercropped crop. So if we focus first on the VALPRO Path project, from the food application side, we had specific targets to adhere to the project. The first target was to actually process the intercrop samples, so that would be the intercrop peas and fava beans, into a flour. But if we're talking about a food application for legumes and intercrops, we need to be very mindful that they do have many anti-nutritional compounds. In particular, they would be high in phytic acid. And phytic acid works by the phytate present in the legumes. They bind to minerals such as iron, calcium, and proteins. And this means that they decrease the bioavailability of these minerals when you eat the food. And that means that we don't really avail of them, or we don't digest them properly. Legumes also contain alkaloids, and when alkaloids are eaten in food, they actually activate bitter sensory receptors in the mouth. So that means that the food that you're eating containing alkaloids are very bitter, and they're very, very unpleasant to eat. And intercropped peas and fava beans would contain high levels of alkaloids. They also contain what we call raffinose family oligosaccharides, or RFOs. These are indigestible complex sugars. So when we eat foods or ingredients containing these RFOs, they can cause very bad bloating and stomach discomfort. So we are at the beginning of the project, we were very conscious that we needed to minimize the anti-nutritional compounds in the intercropped products. And then once we did this, we wanted to produce a flour where the protein content was preserved and also the micronutrient content was preserved, but where the level of anti-nutritional compounds was reduced. And then once we produced a flour, our aim was to focus on novel bakery formulations with a target of a reduced flour substitution by 30%, while at the same time maintaining normal dough handling properties and dough rheological properties, and then keeping the kind of high level of baked product structure and baking performance, and of course, very importantly, maintaining the optimal sensory characteristics of the food that we develop. So this is the experimental design, how we actually processed the intercrops. So as we know, soaking and germination, as it shows in the literature, are a low cost and they're an environmentally friendly practice. Both have shown historically to reduce levels of anti-nutrients in crops. So in our experimental design, we soaked intercropped pea and fava bean for 8 hours and 16 hours. Then we washed the water, then we germinated for 24 hours up to 72 hours. Then we dried the samples, we dehulled them, so we removed the outer layer. We aspirated the samples, which meant we blew away the outer layer, and then we were left with the dehulled fava beans, where we did some two-step milling process and sieved them, and then we obtained a fine refined flour. So the samples that we worked with, where we obtained them from Sheila, thank you very much, straight from the farmers or from the ground. The intercrop samples contained pea and fava bean at levels of 95.5% pea and 4.5% fava bean for one of our experiments, and another intercrop sample contained 85 and 15% fava bean. So once we produced this flour, we needed to analyze the flour to make sure that it maintained good properties and that it could be used as a food ingredient. So we did some microscopy on the flour using scanning electron microscopy. We looked at the approximate composition, the color. Of course, we looked at the anti-nutritional factors of the flour, and then we looked at kind of internal properties such as the starch pasting properties and the rheology of the flour, whether it could make a good dough or a good batter towards a final end product. And we looked at the water holding and the water solubility capacities. Now, we can't claim all of the credit for all of this work because we had some really nice collaborations within the VALPRO project. So thank you to those partners from Spain, from Denmark, and from Athens in Greece who helped us with some of the analysis. If we look at some of the results then, as expected, we found that soaking significantly decreased the level of phytic acid of the intercropped products. Also germinating decreased as well, especially for the RFOs, the raffinose and the stachyos. We found with 72-hour germination, there was a very significant reduction of stachyos and a complete elimination of raffinose. And this is because RFOs are actually water soluble. So when they're soaked, the actual RFOs leach from the grains and into the soaking water. So it's really important when you are soaking and before germination that you throw away the soaking water because it actually contains the RFO carbohydrates. Then for germination, as I said, we found that there was a significantly reduced level of phytic acid. And what happens during germination is that an enzyme called phytase is activated, and this breaks down the phytic acid, and it actually releases the minerals such as the iron or the calcium that the phytase binds to. So these are now released and they're available for digestion and to be absorbed within the consumer when you eat the food. Then something that we were also very wary of in the flour, because it's predominantly pea with some fava bean. Pea is green, therefore the pea flour is going to be green, or the resulting flour is going to be green. So we were also very pleased to find that with germination, the A, which is a measure of the green undertone or the green color of the flour, was significantly reduced. And this is because during germination, the chlorophyllase enzyme is activated, and this actually cleaves the chlorophyll molecule, and it means that the color becomes much less green and much whiter in comparison to a normal white flour. Okay, so then we had this flour. It had a reduced anti-nutritional compound. It maintained protein and fiber levels. So what we wanted to next was to bring this flour into some end products. So we looked at a sweet cake formulation where we actually replaced the wheat flour by up to 50% using intercrop pea and fava bean at 94.5 and 5.5 ratios. We formulated the sweet cake, we replaced wheat flour, and we looked at the rheological properties, composition, the internal structure of the cake, and also the anti-nutritional compounds. And all the time we were comparing to a wheat-based control formulation because we didn't want it to significantly differ from something that people would normally be eating day to day. So we looked at sweet cake. We've also looked at a cracker formulation where we also replaced wheat flour up to 50% using pea and fava bean at 85 and 15% ratios. And same again, we looked at the dough textural properties, the crispy versus the crunchy properties of the cracker over an 8-week staling period. We looked at the kind of the water activity, how it's staled, how the starch retrograded over time. And we are currently doing the same now with a soda bread formulation, and just the pictures underneath are some of the products that we have formulated. So if we look first very quickly at some of the sweet cake results, you can see pictures of them there. These are where we have reduced or substituted wheat flour from 30 up to 50%. And what we found, as expected, was that there was a significantly higher level of protein in the intercrop substituted cake. But interestingly, there was an unchanged specific volume at 50% inclusion levels, which meant that it was maintaining the same baking structure, maintaining the same volume, the same shape, when baked. Also, there was a similar rate of crumb moisture loss. So you probably know when cake or bread stales, moisture migrates from the center of the product to the outside, so the crumb becomes dry and hard and stales. However, we found that the rate of staling over 7-day staling period was significantly not different from the control wheat formulation. So they actually staled at the same rate as a wheat control. So in the previous slide, I showed the moisture migration. This slide actually shows the crumb hardness using texture profiling. And you can see again, over the 7-day staling period, the cake with the 50% level of inclusion actually staled at a similar rate to the wheat control cake. Also, we did some 2D structural imaging using a C-cell machine. This actually showed, which was surprising, that the more fava bean or the more intercrop flour that we introduced into the cake formulation resulted in a more uniform crumb grain. So there was less large air cells. There was a better, more kind of uniform structure to the inside or the internal grain. And also the RFOs of the cakes, or the oligosaccharides containing the intercrop flour, was comparable to the control. So despite the fact that we were introducing intercrop legumes, which do contain levels of RFOs, it just shows that through the soaking and the germination processing, we were able to reduce them to a level where they were introduced. It didn't significantly change their levels when compared to wheat. These are just some of our cracker results. Again, there was significantly high content of protein in the end product in the crackers when we substituted with 25% and 50% of the intercropped. There was also a higher product density. So because there's a higher protein content and also a higher density, it means that the product would have more of a satiating effect. So you would feel fuller for longer. So we would deduce from that, when you eat 1 cracker or 2 crackers, you might feel a lot fuller than when you would eat a normal wheat cracker and you reach into the pack and you have another 8 or 9 crackers to try and feel full. So that's quite an interesting result. And also the staling profile resulted in the crackers becoming a little bit more crunchy rather than the crispier wheat products. So that would also help with the feeling fuller for longer once you've eaten. So there are 2 products. The ongoing work that we're doing with intercrops is also including not a yeast bread, but a soda raised bread trial. And we'll also be working on a 4th product before the end of the VALPRO project. Once we've optimized all of the product formulations containing the intercrop, we'll be doing food safety trials, of course, and then we will be doing larger sensory acceptability of all of the optimized products. But I should say that our in-house panel are doing some basic sensory trials throughout our experiments, just to make sure that we are producing some acceptable products to date. Once all our products have been optimized, we'll be disseminating our results through scientific and also technical publications. So that's some of the work that we've been doing on intercrops with fava bean with pea. This is another project that I'm going to speak about really quickly. This is a project called G-Free IE. It's a project that's being funded by the Irish Department of Agriculture, Food and the Marine. It's where we're focusing only on Irish fava bean as a gluten-free ingredient to formulate novel high protein, high fiber, gluten-free products. So similarly, we are doing some similar processing of the fava beans. So we're still going to soak them and germinating them. We're going to look at reducing the anti-nutritional compounds. And then we are also at the moment innovating for the whole gluten-free market. So we're developing gluten-free bread, puffed or extruded snacks, and also flaked breakfast cereal products. At the same time as this, our partner in Moorpark is looking at celiac gut studies. So we're making sure that the gluten-free products that we do formulate are able to the celiac gut will be able to still absorb the nutrients and the proteins in the products that we do formulate. And also we will be doing a life cycle analysis throughout this project as well, so that project is also underway. These are just some of the products that we have formulated so far in the gluten-free project. On the left is a gluten-free bread containing Irish-grown fava beans. We're also combining with quinoa and with Irish oats. The project, or the products in the middle, is Irish-grown fava bean that have been extruded, so to make extruded snacks and crisps. And then the product on the right is where we are using a flaking process to make some new gluten-free breakfast cereal flakes, and this picture is just fava bean on its own, but again we'll be combining with oat flakes and other gluten-free maybe proteins as well, and then we'll be optimizing the sensory properties of all of these by the end of the project. So just to conclude my presentation, as we see, new crop science approaches are happening at the moment, which is good to see, with a focus maybe on human health and new food innovation. If you are working with legumes or high protein crops, it's also important to be aware of the anti-nutritional properties that legumes do have, and how maybe green processing and low-cost processing are very effective to reduce the anti-nutritional compounds of these legumes. Then also, when you're working with high protein crops, it's important to match suitable applications with the correct products. And to date, maybe our food innovation trials have shown that intercropping with fava bean and fava beans on their own can show potential for, I suppose, for agriculture, for the environment, also for sustainable practices, and then at the end of the road for a new product or for new food applications. And then just before I finish, I'd like to acknowledge my colleagues who have done a lot of this work. So there's Dr. Miraj Fatima Mullah, Dr. Marianne Matrens, and Mr. Patrick Keys. Okay, and that's my presentation. Thank you very much.
No, thank you, Dr. Mir. Again, we have discovered there are multiple factors in creating plant protein foods, and thank you for explaining further. Now we are coming to our Q&A session part. If you have any questions for any of our speakers, please, you can write it down in the Q&A and we'll answer them. There are a couple already questions, so we can go to that. First one, a little bit comical, but let's say, did I hear correctly that the pigeon attacked a grown pea plant? So I think this was part of Sheila, your presentation when the...
Yeah, that's right. Now they don't attack the plant, they attack the pods, especially the ones that are chattered, and they remove the peas. They do attack plants when they are pea plants when they are small. They graze on the leafless leaves of the plant, and it actually because they are so small, the pea leaf plant, that it can impair actually the growth of the plant, but that's at another stage. At harvest, it's the shattered pods that they will go after first.
Yeah, thank you. Thank you for answering, Sheila. And now we have one. It's how can, well, I presume it was about the research done in that Professor Mark was doing. How can we use that research for yogurt, and what process can be used to reduce the convicin and vicin for people with favism? I
Yeah, I
think,
can address those two. So
yes.
just in terms of the yogurt, yeah, it can be used. It's a very suitable alternative for maybe soya protein. So the fava bean isolate is very useful for that. It actually produces a very creamy texture after fermentation. It's not something we would have done in VALPRO Path, but yes, fava bean can be used in both concentrate and isolate form in yogurt production. And then the second one on the vicin, which is obviously a contentious issue with favism, a very important disease worldwide. The levels of vicin and the current breeds or varieties of, and Sheila, you can jump here if you want, of varieties of faba bean is actually very low. And we found that, or it's proven that with a lot of the treatments that faba beans go through when it's put into a food application, such as heat treatment and so on. It can actually knock off these anti-nutritional factors, and two of these are the voicing co-voicing element of this. So with breeding and processing, we can eliminate this effect.
Exactly. At the moment, there are varieties classified as very, very low vicin and covicin, and they were bred specially for that trait. They are not zero like other anti-nutrients like tannins, where you can have zero tannin-free varieties. You cannot have that with faba beans, but as with vicin and covicin in faba beans. But as Richard said, throughout the process of extracting the protein and other ingredients of interest, a lot of the vicin and covicin is also removed.
All right. Thank you, thank you, Dr. Ritter and Dr. Sheila, for the answer. We have one for Sheila. What future research is required to improve the variational robustness of Faba bean in Europe?
I think we have to start selecting the varieties for the different regions and map accordingly. The other aspect on top of that is the climate changes. So a variety that is growing very well in Ireland at the moment might not grow so well in a couple of years because it will become more dry, for example. So it's important to have, I suppose, a repository of the varieties and their traits, so we can prepare better for climate changes and so on in the future.
Thank you, Dr. Sheila. We have another question here. What was the intention behind using wet milling instead of dry milling for the fava beans? Are there any benefits of milling in a wet system? I think
In
this
terms of
is
the
one.
processing side of things, the wet milling has been done as a pre-processing step to put it into solution. And the idea of that is that you can separate, I suppose, at the moment, the dry fractionation and wet fractionation focus specifically on the protein element of the faba bean. But what we're trying to do is valorize the full plant itself. So we take the starch and fiber components of that as well and get value from that. So by wet milling and putting it through the different separation protocols, then thereafter with separators, we can separate the separate side streams or the categories of components in the faba bean and then valorize those so we can sell the starch as a raw ingredient or fiber and so on.
Thank you, Dr. Richard. Now we have one more question for Dr. Eimear. What do you see as the biggest driver for inclusion of legume flavors in bakery applications?
Well, there would be multiple drivers. I suppose the main driver would be that it's healthy, it's high in fiber, it's high in protein, the fats are unsaturated, it's high in minerals. Once they're processed, it really enhances the bioavailability of all of these nutritive compounds. They have the ability to reduce the GI response in foods when added. And then if we work backwards from the food, as we've seen from Sheila and from Richard, that they're quite a sustainable crop from the growing size, that they need less artificial fertilizers. So I suppose from the field right through to the fork, there would be a lot of both agronomy and food health advantages to these crops.
Oh, okay. Thank you, Dr. Eimear. We have more questions here. So, hi everyone. Many thanks for all the highly interested presentations. I have a question for Dr. Gallagher about the process of faba bean flour production. The step of germination, develop the germ. Is it removed or milling with a whole grain after grinding?
It's milled. So they're dehulled to remove the outer, the hard outer coat, and then they're milled
Okay.
and sieved. Yeah.
Yeah, thank you, Dr. Gallagher. So we have, well, I think one more question. Then we should already be ending the webinar soon. Do you think legumes other than pea and faba bean should also be considered for intercropping? I think this is for Dr. Sheila.
Yeah, so there's other all depends on the location, right? So here we can only grow cold season legumes, so faba beans, peas, and lupins. In the southern Europe, they can do chickpea and lentils and so on. And there's absolutely no reason why we cannot intercrop any of them with other legumes, but also with other cereals. It depends on the goal. What's the aim of intercropping? Is it higher productivity, support for one of the crops with suppression? I think according with the goal, you decide on the species to be in, to add for the intercropping.
Right. Thank you, Dr. Sheila. And I think I will take one more question. What are the market opportunities in Europe today for faba beans? Are there already any companies using faba or faba flowers? I think, Richard, you could.
Yeah, I can address that. I see Eimear's not jumping on that one. So,
I was just about to.
so yeah, so in terms of obviously there's a growth in a plant-based food market in Europe substantially. Traditionally that would have been met by the pea protein because it's a very stable and it's a neutral tasting protein in terms of plant. But faba bean obviously because of what we said is a very versatile crop, grows in a lot of regions and yields very well. So we're trying to see if we've outputs for this crop. In terms of faba bean flour, at the moment we produce very little. In Europe, we import most of our faba bean flour that's been produced and used in the food manufacturing industry. So there's definitely a market for that. For the concentrate and isolate, there are a number of facilities that have set up and have started producing concentrate and isolate. It's an expensive process, and what they tend to do is they tend to bring in other crops with that to try and make the facility economically viable. But there is definitely a growth in the market for plant-based products, food offerings, and I think faba bean can definitely can fill this void.
I can add to that the snack industry is also looking a lot to include and substitute things used before, like potatoes, for example, with protein crops or snacks like salted and roasted faba beans or lentils puffs and things like that. We see it more and more, and they are done in Europe too.
Oh, thank you, Dr. Richard. Thank you, Dr. Sheila. I think with this, I think the more questions, if they are, you can always contact us at the VALPRO Path through our website or through our social media channels that we have on LinkedIn, Facebook, and Twitter. And if you have any more questions regarding any of Faba related plant protein information, you can always, as I said, contact us and we will be sure to answer as soon as possible to all of your questions. With this, I would now be ending our webinar on creating plant protein value chains for faba beans. As Dr. Richard already said at the beginning of the webinar, we have much more coming in the upcoming weeks, months, with the next webinar being on Monday, the 13th of April, that will be on chickpeas. So if you're interested, we already have on our social media channels a link to register for that. I would once again like to thank Dr. Dr. Sheila Alves for being part of the webinar, Dr. Eimear Gallagher for being part of the webinar, Dr. Richard Lynch for being a great co-moderator and part of the webinar, and Professor Mark Fenelon as he had to leave for a different meeting. But once again, and thank you all for attending. If you again have any more questions, you can always contact us, and that will be all. Thank you.
Thank you.